Triple sugar iron agar. General. Whenever you see the name of this test, triple sugar iron agar, remember that it's a test involving 3 sugars: lactose, sucrose, and glucose, and iron. It also contains agar as a solidifying agent, making it a semi-solid medium with a slant and a butt. Composition of triple sugar iron agar. Triple sugar iron agar contains lactose, sucrose, and glucose in a 10-hour, 10 minutes, and 1 second concentration ratio: 1% lactose, 1% sucrose, and 0.1% glucose. 0.1% glucose. If only glucose is fermented, only enough acid is produced to turn the butt yellow. The slant remains red. 1.0% lactose per 1.0% sucrose. Fermentation of lactose or sucrose or both produces a large amount of acid, turning both the butt and slant yellow. This indicates the isolate's ability to ferment lactose or sucrose or both. Iron, ferrous sulfate, an indicator for hydrogen sulfide, H2S formation, appearing as a black precipitate, phenol red. A pH indicator that turns yellow in acidic conditions and red under alkaline conditions. Peptone provides a source of nitrogen. Aerobic utilization of peptone leads to ammonia production, potentially causing a deeper red-purple color on the slant. Why sucrose is added? Sucrose is included to allow for earlier detection of coliform bacteria, which ferment sucrose more rapidly than lactose. It also helps identify certain gram-negative bacteria that can ferment sucrose but not lactose. The tube structure, with a poorly oxygenated butt and a well-oxygenated slant, aids in differentiating bacterial metabolic capabilities. Triple sugar iron agar test procedure. 1. Using a sterilized straight inoculation needle, touch the top of a well-isolated colony. 2. Inoculate the agar by stabbing through the center to the bottom of the tube, and then streaking the surface of the slant. 3. Leave the cap loose and incubate at 35 degrees Celsius in ambient air for 18 to 24 hours. Interpretation of triple sugar iron agar test. 1. Lactose/sucrose fermentation. A large amount of acid is produced, turning both the butt and slant yellow. Gas production may cause bubbles or cracks. 2. Glucose fermentation only. The oxygen-deficient butt turns yellow, while the slant remains red due to acid oxidation. 3. No fermentation. Both the butt and slant remain red. Ammonia production may cause a deeper red-purple color on the slant. 4. H2S production. A black precipitate of ferrous sulfide is observed. Key interpretations of triple sugar iron agar test. Red/red. No fermentation of glucose, lactose, or sucrose. Red/yellow. Glucose fermentation only. Yellow/yellow. Glucose and/or lactose sucrose fermentation. Black precipitate. H2S production. Bubbles/cracks. CO2 or H2 production. History. The triple sugar iron agar test is based on a medium described in 1917. Hajna's 1945 modification, incorporating phenol red, is the formulation still used today. Poem on triple sugar iron agar test. A test of 3, a sweet design, lactose, sucrose, glucose in line. Iron's gaze, a sulfide's hue, in agar's hold, reactions brew. A slant, a butt, a world apart, where microbes dance, a work of art. Yellow or red, the colors speak, of sugars tamed, and gases leak. From ancient roots, Hajna's hand, a tool to know, to understand. Salmonella's dance, Shigella's sway, in triple sugar iron's display.